Gene therapy. What is gene therapy? A gene is a unit of hereditary and carries inherited information. Here we can find the gene on the chromosome formed of exon and intron followed by exon. Some facts about gene: 3 billion letters in DNA code in every cell in our bodies. There is 6 feet of DNA in each of our cells, packed into a structure of only of 0.004 inch across. It would easily fit on the head of a pin. There are 100 trillion cells in the body. If all the DNA in the human body was put end to end, it would reach the sun and back over 6 times. 100 trillion x feet individual by 93 billion miles. If we resetted the genome at 1 liter per second for 24 hours a day, it would take a century to reset the book of life. The vast majority of DNA in the human genome, 97%, has no known function. Our DNA is 98% identical to that of chimpanzee. Between human and our DNA differ only by 0.2%, 1% in 500 bases later. This takes into account that human cells have 2 copies of the genome. To estimate number of genes in both human and mice is 30,000 to 45,000. In the roundworm C. elegans, the number is approximately 90,000. Gene. What's gene exactly? When the gene speak about himself, he say, I am carried on a chromosome. I am the basic unit of hereditary. Encode how to make a protein, DNA, become RNA, become proteins, and central dogma. Protein carry out most of life function. When altered, causes dysfunction of a protein. This number of genes per chromosome. Gene structure. We find the gene formed from promoter, exon 1, exon 2. How function is done: transcription, splicing, and translation. We get the protein function. Our life is maintained by a molecular network system in the cell. Protein plays a key role in the living system. 3 examples of protein function: catalysis. Almost all chemical reactions in the living cell are catalyzed by protein enzyme. Transport. Some protein transports various substances such as oxygen, ion, and so on. Information transfer, for example, hormones. Functional genomics, from gene to function. Genome expressed and called expressome. Proteome, protein is formed, metabolite on the other level. When there is a mutation in the gene, then it will change the codon, which will change which amino acid is called, for which will change the conformation of the protein, and this protein function will be altered, and the genetic disorder result, and this is from mutation in the genome. Imagine, for example, that you accidentally broke one of your neighbor windows. Let us imagine with each other what will you do. Number 1 action: stay silent, or repair it with some tape, or put in a new window. what is similar in gene therapy: stay silent, ignore the genetic disorder, and nothing gets fixed. Try to treat the disorder. The number 2 action: try to treat the disorders with drugs or other approaches depending on the disorder. Or the 3, put in a normal functioning copy of the gene. If you can do this, it will solve the problem. How to fix it? Virus and modified virus. A virus is found. We use the virus as a vector. We use the virus as a vector to transfer a gene. A virus is found which replicates by inserting its gene into the host cell's genome. This virus has 3 genes, A, B, and C. Gene A encodes a protein which allows this virus to insert itself into the host genome. Gene B and C actually cause the disease this virus is associated with. Replace B and C with a beneficial gene. Thus, the modified virus could introduce your good gene into the host cell's genome without causing any disease. So we use the modified virus to fix the broken window. Gene therapy using adenovirus vector. New gene is inserted into an adenovirus vector which used to introduce the modified DNA into a human cell. If the treatment is successful, the new gene will make a functional protein.